Gene therapy and genetic manipulation, the future of cancer treatment. Modern cancer research has found new and promising treatment options for various types of cancer, and even better, they are invasive and have minimal adverse side effects in comparison to current treatment options such as chemo and radiation. Through immunomodulation of both the tumor cells and the host immune system combined with certain alterations to the microenvironment of the tumor itself, researchers have found that the immune system can be taught to recognize and attack cancer cells and force regression of tumors. This treatment process involves a combination of gene therapy and manipulation of host genes to accomplish, and employs a myriad of highly advanced methods, 6 of which I will address today. I personally have a very long and painful history of cancer in the family. I've seen many different kinds of cancer firsthand, as well as their treatment approaches. With these are often traumatic side effects and harrowingly low success rates. However, now that gene therapy research is starting to truly show promising data, it's coming under scrutiny, but not by scientists. Due to widespread misinformation, many religious groups as well as some more conservative individuals with social media influence, setbacks in research and strict rules are preventing the research from advancing and being available to the patients who need it most. In compiling and providing factual information, I aim to educate others on the effectiveness of this treatment and the importance of the research being done. Many methods currently exist within the world of gene therapy, but the following have proved to be the most effective and promising. Small interfering RNAs can be engineered and employed to disrupt protein-gene interactions within cancer cells. These interactions are necessary because they manufacture proteins, which are used as energy sources and structural sources for cells, and without them the cells die. The next is nitrogen and phosphate manipulation in plasmid DNA vectors. The ratio of nitrogen and phosphate within the cell directly influences gene expression and efficiency of gene transition and translation. By altering this ratio, scientists are able to tailor the way in which DNA retrieval phosphate groups deliver DNA to the necessary location for them to be expressed. By tailoring this ratio a certain way, scientists have been able to suppress genes in cancer cells that contribute to the rapid and nonstop growth. The third is hybrid carbon-based materials. One of the biggest clinical challenges of gene therapy today is the way in which the genes are delivered to the treatment site within the body, which is called the target cell. New advances in nanotechnology centered around a molecule that is natural to all living life forms, which is carbon, can guarantee a much safer delivery to the patient and a more safer environment for the practitioner than those used before, such as viral delivery. Anti-angiogenesis therapy, using monoclonal antibodies, MABs, in order to block signaling that induces angiogenesis within cancer cell groups has been approved by the FDA and is very effective in drastically slowing the progression of cancer cells, and this allows for treatment of highly aggressive cancers. Cytoreductive gene therapy and editing. Maxillofacial and oral oncologists are currently researching ways to employ gene therapy on not only solid tumors, but tissues that have a high chance of becoming tumorous. By using gene editing, they are reducing the chances of cancer spreading throughout the head and neck via the available tissue. This allows oncologists to keep the tumor centralized, making them more easy to remove, but also serves to make radiation and chemo treatment applied to the cancer less damaging to the rest of the body. The last one is immunomodulation of gene host cancer cells. By using viruses armed with genetic material that alters the homeostatic mechanisms of cell bodies, researchers have found a way to make unfavorable conditions to cancer cells that do not affect the other cell types. This treatment has proven infinitely valuable in maintaining regression of cancer after the initial successful removal and treatment. Many of the gene therapy techniques employed today are void of the negative side effects, often associated with current and more traditional treatments. 90% of the cells targeted via the cytoreductive gene therapy are cancer cells that contribute to tumors forming within facial tissue. This leaves most other natural, healthy cells untouched and undamaged. This helps escalate recovery rates and guarantees the ability for nearly full natural tissue reconstruction after the cancer recedes. The siRNA treatment allows a myriad of major organs to be directly targeted without damaging surrounding organs, or even risking damage on any tissue not in direct